Hi, I'm Katarina Quinlan. I'm an assistant professor of biomedical and pharmaceutical sciences at the George and Ann Ryan Institute for Neuroscience at the University of Rhode Island. My lab studies electrophysiology and morphology of neurons before the onset of neurodegenerative disease. In a recent study, we looked at inhibitory interneurons in the spinal cord from a mouse model of ALS. Patch clamp revealed very different intrinsic properties of ventral glycinergic interneurons, particularly a more depolarized onset and peak voltage of the persistent inward current of the glycinergic interneurons from SOD1 mice. As a result, inhibitory interneurons from SOD1 mice were found to have significantly higher threshold than wild-type interneurons. This means that the interneurons were less excitable. In vivo, glycinergic interneurons in SOD1 mice probably fire less than the same interneurons in wild-type mice. In order to determine a little bit more about the identity of these interneurons, we performed a regional analysis. We divided the ventral horn into the ventralmost 100 microns, the area where Renshaw cells are found, lamina 9, the motor nuclei, and the rest of the ventral horn, consisting of lamina 7 and 8. The most dysfunctional interneurons were found in the ventral region, where Renshaw cells reside. This study shows for the first time that inhibitory interneurons, likely Renshaw cells, are impaired in excitability in ALS. This could have functional consequences that lead to loss of inhibition of motor neurons, and even could be used as a potential biomarker for ALS. Clinically, this is important because drugs like riluzole that universally reduce excitability could have a more mixed effect than we had previously thought, because it would be reducing excitability of these inhibitory interneurons that we show are already impaired. This paper is an editor's choice pick in issue 601.3 in the Journal of Physiology. Thank you for taking the time to learn more about my work.